once the samples have been drying for about 30 minutes, they should be ready. Bob is now going to add 500 microlitres of distilled water to each tube.
Yep, so what we do is we just take the tubes, the dry genomic DNA samples, we adjust our pipette to 500 microlitres, so 0.50 mils or 500 microlitres, and then we add 500 microlitres of deionised water to each sample. Sorry about that. And what we do is we just gently suck up the solution just to help resuspend that genomic DNA in solution. Okay.
So rather than vortexing, Bob is just going to gently pipette that solution up and down to resuspend the genomic DNA, otherwise known as gDNA.
It's important to also note that deionised water, or the distilled water that we're using, has actually been sterilised or autoclaved to make sure there's any contaminants in it, because there's no point in having any contaminants in that water which may then interfere either with your PCR reaction or actually contaminate the genomic DNA and actually degrade it. So we're actually using sterile deionised water. And now we have it. So here we've got our two genomic DNA samples from wheat plant 1 and wheat plant number 2. And what we have to do as good standard practice is include what it is. So we just write genomic DNA on the side here, W1, and today's date, which is the 16th. Okay, and so we'll actually put that in our notebook of what we did today, so therefore we know if we had to refer back to our notes, we know what each of these tubes has in it.
Okay, so our genomic DNA from wheat is now extracted and ready to use in downstream applications such as the polymerase chain reaction or PCR. Thank you, Bob.
So thank you, Barrett.